Welcome back to the deep dive. Today, we're tackling a really cool story from biology. It's about how scientists found this glitch in the rules of inheritance. And instead of being a problem, it turned into this amazing tool, like a genetic ruler.
Yeah, it's fascinating. You start with Mendel, right? Everyone learns about Mendel.
Right. His second principle. Independent assortment. Everything's supposed to be neat and tidy. You cross two dihybrids, you expect that classic 9.3.3.1 ratio, like eye color and hair texture, shouldn't influence each other.
That's the theory. But, well, almost right away, researchers like Bateson and Punnett, they started seeing things in the real world that didn't fit. Some traits just seemed stuck together. They weren't getting that 9.3.3.1 mix at all. Way too many offspring looked just like the parents.
Okay, so not independent then.
Exactly. And that leads us straight to linkage. It's just the physical reality.
Yeah.
Genes on the same chromosome tend to travel together during meiosis. Think of them as being on the same bus.
Ah, okay, linked together.
But here's the twist, and this is really the core of our deep dive today, that linkage isn't perfect. It's not absolute.
Right, and that's the revolutionary part, isn't it? If they're linked, but sometimes they separate, how do you even measure that? So our mission today, to figure out how T.H. Morgan and his student, Alfred Sturtevant, use this imperfect linkage to actually measure the physical distance between genes, to map the chromosome.
It's a fantastic story. Morgan was the fruit fly guy, Drosophila.
Yes. Everyone remembers the fruit flies.
He saw clear evidence of linkage. But his big insight was why they sometimes separated.
What was it?
He figured genes must be arranged linearly, like beads on a string on the chromosome. So they sometimes came apart when they shouldn't have. It had to be because of some kind of physical break and swap.
A physical exchange.
Precisely. He called it recombination.
Okay, so Morgan spots recombination, but Sturtevant takes it further.
Oh, way further. Sturtevant had this brilliant idea. He looked at Morgan's fly data, all those percentages, and he thought, what if the frequency of recombination? How often they swap.
Yeah.
What if that frequency tells you the distance between the genes? More swapping means they're further apart. Less swapping, they're closer.
Wow. So like the randomness of the swap location actually becomes a measurement.
Exactly. It's genius. He realized this biological glitch was actually a ruler hidden in the cell.
That's incredible. Okay, let's make this concrete for everyone listening. Think about a standard test cross, maybe AB crossed with the recessive A.
Perfect example. If those genes A and B are unlinked, totally separate chromosomes.
Then you get Mendel's prediction.
Right. 4 kinds of offspring, all in equal numbers, 25% each. Simple probability.
But if they are linked?
Now it gets interesting. If they're linked, most of the offspring look like the original parent combinations because the alleles on the bus stayed together.
Those are the parental types.
Yep. And then you get a smaller number, maybe just a few percent,
Yeah.
that have new combinations.
Yeah.
Those are the recombinant types.
And they only happen because of that physical swap, that recombination?
That's the only way. The frequency of those recombinants tells you the story. More recombinants means the genes are farther apart on that chromosome string.
Okay, so the genes want to stick together because they're on the same chromosome. Let's unpack the physical process that actually breaks them apart. What is it?
It's a process called crossing over, or you might hear it called homologous recombination. It's this really precise molecular dance that happens when sex cells, gametes, are being formed.
In meiosis, right?
Specifically, prophase meiosis. This is when the chromosomes that you got from your mom pair up really tightly with the equivalent chromosomes from your dad. They form this structure.
The tetrad.
The tetra, exactly, or sometimes called a bivalent. 4 chromatids all bundled together. And that's when they can physically exchange segments, swap pieces of DNA.
And the actual spot where they touch and swap, that's the chiasma.
That's it, the chiasma, plural chiasmata. Where that chiasma forms is, it's the whole ballgame, really. Well, think about 2 linked genes, A and B. If a chiasma forms between A and B, the swap will separate them. You'll get recombinant gametes.
Okay.
But if the chiasma forms somewhere else on the chromosome, outside that A to B region, then A and B will stay together on the same chromatid. No recombination between them.
So being close together physically protects them from being separated by a random crossover event.
Precisely. If genes are super close, the chance of a chiasma landing right between them is low. So you see very few recombinants. They have tight linkage.
Makes sense.
And the flip side, if genes are far apart on the same chromosome, there's just more room, more physical space for a random chiasma to land between them.
So you get more recombination.
You get a higher frequency of recombination, yeah. more recombinant offspring. The distance dictates the probability.
Okay, quick detail here. When this crossing over happens in the tetrad, that four-strand structure, does it involve all four strands swapping parts?
Oh, good question. No, it's actually more specific. At any single chiasma, the physical exchange only involves two of the four chromatids.
Only two.
Yeah, usually one chromatid from each homologous chromosome. That's why even when crossing over happens, you still get parental-type gametes produced from that same meiosis event, alongside the recombinant ones.
Right, because the other two chromatids weren't involved in that specific swap? Okay, got it.
So we've got these observations, these percentages of recombinant offspring.
Yeah, how do we actually, turn that into a map, a standardized measurement?
It comes back to Sturtevant's core idea. Chromosome mapping is basically just translating that recombination frequency directly into distance.
How's the calculation work?
It's surprisingly simple, actually.
Yeah.
Just count up your recombinant offspring.
Okay,
Divide that number by the total number of offspring you looked at, parental plus recombinant.
right. The proportion.
Then multiply by 100 to get a percentage. And that percentage is the map distance. We call the units map units.
Or centimorgans, right.
After Morgans. Exactly, centimorgans, cm. One map unit equals 1% recombination frequency.
So, let's say we did a cross, counted 400 total babies, and 100 of them were recombinant.
Okay, so 100 divided by 400 is 0.25. Multiply by 100.
25%. So the genes are 25 map units apart.
Precisely. 25 map units or 25 centimorgans. It's that direct.
But you mentioned a caveat earlier, a limit.
Ah, yes. There's a ceiling. The maximum recombination frequency you can directly measure between two genes is 50%.
50%. Hold on, that sounds familiar. Isn't that what you get with unlinked genes? Independent assortment?
Exactly. If two genes are really, really far apart on the same chromosome, or if they're on different chromosomes entirely, they'll assort independently. You get roughly 25% of each of the four possible gamete combinations.
Which means a 50% recombination frequency overall, 2 recombinant classes out of 4 total classes.
Right. So if genes are 50 map units apart or more, they behave as if they're unlinked in a standard cross. Crossing over is essentially guaranteed to happen between them somewhere.
So we can't map distances beyond 50 map units using just one cross like this.
Not directly, no. We'd need to use intermediate genes, kind of like stepping stones, to map longer distances. But for any pair, the observable limit is 50%.
Okay, that makes perfect sense. Let's tie this all together with that rabbit example from the sources. Genes A and L.
Good idea. Real data always helps.
So we have this cross, a dihybrid female rabbit crossed with a homozygous recessive male. We're tracking 2 traits, spot pattern and hair length. A is unspotted, A is spotted, L is short hair, L is long hair.
And crucially, the results don't look like independent assortment. The numbers are skewed.
Right. The data showed 75 spotted short-haired rabbits, 66 unspotted long-haired ones. Those are the big numbers.
And then only 10 unspotted short-haired and 7 spotted long-haired. The small numbers.
So the big numbers, 75 and 66, those must be the parental types, right? They tell us how the alleles were linked in the dihybrid parent.
Exactly. Those high-frequency classes reveal the original arrangement on the chromosomes in that female rabbit.
And the small numbers, 10 and 7, those are our recombinants, the result of crossing over.
That's right. They have the new combinations of alleles that weren't present on the parent's original chromosomes.
Now, you mentioned something important when we prepped the configuration, cis versus trans. Can you explain that again? Why does it matter here?
Absolutely critical. The configuration tells you which alleles started out linked together on the same chromosome in the parent.
Okay.
If the parent was cis, it means the 2 dominant alleles, A and L, were on one chromosome, and the 2 recessive alleles, A and L, were on the homologous chromosome, like ALL.
Got it. Dominants together, recessives together.
But in this rabbit example, look at the parental offspring, the big numbers. One is spotted short, AL, and the other is unspotted long, AL.
Ah, so a dominant allele of one gene is linked with the recessive allele of the other gene.
Precisely. That's the trans configuration, a linked with L or linked with L. So the parent's genotype regarding these linked genes was effectively AL.
And knowing that configuration is key because it tells us that the other combinations, the unspotted short AL and spotted long offspring, the ones with counts of 10 and 7, must be the recombinants.
You got it. If the parent had been cis, allal, then those 10 and 7 rabbits would have been the parental types and the 75 and 66 would have been the recombinants. It flips everything.
Okay, that's a crucial distinction. So knowing it was trans, Our recombinants are the 10 and the 7.
Right, so let's do the math.
Total recombinants is 10 plus 7 plus 17.
Yep, and total offspring, it gets 75 plus 66 plus 10 plus 7. That's 158.
Okay, so the calculation is 17, 158, 100.
Exactly. 17 divided by 158 times 100.
This gives us 10.8.
10.8 mapped units. That's the calculated distance between gene A and gene L on the rabbit chromosome.
Just by counting rabbit fur and spots, that really is amazing. We've measured a physical distance inside a chromosome.
It's incredibly powerful. And it means we can summarize the possibilities for any two genes in a dihybrid cross really neatly.
What are the scenarios?
Well, first, you've got the classic Mendelian situation. Genes are on different chromosomes. They assort independently. You see that 9.3.3.1 ratio in a dihybrid cross outcome, or 1.1.1.1.1 in a test cross. Easy.
Okay, scenario 1.
Second, you could theoretically have genes so close together, like right next door, that crossing over almost never happens between them. They're tightly linked.
So mostly parental types then.
Almost exclusively parental types. It would look more like a 3.1 ratio if you did a dihybrid cross, because the linked unit acts like a single gene. Very rare to be that tight, but possible.
Right. And the third?
The third is what we just explored. The genes are linked on the same chromosome, but they're far enough apart that crossing over does happen between them at some measurable frequency.
Like our rabbits?
Like the rabbits. And the offspring ratios won't be 9.3.3.1, nor will they be purely parental. They'll be somewhere in between, dictated by that recombination frequency, that map distance, like the 10.8 map units.
It's just a fantastic illustration of using probability in biology. Observing these patterns lets us build a physical map without needing, a super high-powered microscope to trace the DNA itself.
Absolutely. And we often talk about recombination mainly for its role in evolution, shuffling genes, creating diversity, which is vital.
Sure.
But this mapping aspect, it shows recombination is also this built-in measuring tool. It reveals the chromosome's physical layout.
Yeah.
So here's a final thought to chew on. Since recombination frequency depends on distance, what if you have a set of genes that work really well together, giving a big survival advantage?
Okay.
What happens if, through evolution, those advantageous genes end up really, really close together on the chromosome? Maybe less than one map unit apart.
Recombination would almost never split them up.
Exactly. They'd become effectively locked together, inherited as a single block, a supergene.
So selection wouldn't just act on one gene, it would act on the whole beneficial package deal.
Precisely. That physical proximity, that lack of recombination, could become a powerful evolutionary force in itself, preserving successful combinations. The map distance itself has evolutionary consequences.
Wow, that physical spacing is like an unsung hero of evolution. Something to think about. Well, that's all the time we have for this deep dive. Thanks for joining us.